Okay, so today's video is going to review some of the experiments we talked about in class today, and then we're going to flip over and focus on structure of DNA and how it replicates. So the first experiment, I just want to put emphasis on a couple of the ones we talked about today. And so the first one being Griffith's experiment. So this was the experiment with the bacteria. So in essence, he worked with two kinds of bacteria. He worked with what were called S, or smooth cell bacteria, and R, or rough cell bacteria. And so this referred to the protein, the polysaccharide coat that surrounded these bacteria. And the smooth polysaccharide coat protected the bacteria from the mouse immune system. So it was then able to survive and infect the mice. The rough polysaccharide coat, the immune system would recognize that, and it would the immune system would recognize that, and it would be rendered harmless. Okay, so when the living smooth cells were injected into the mice, the mouse would contract pneumonia and the mouse would die. And when the living R cells were injected into the mouse would survive and it would be fine and healthy. So Griffith then took S cells that had been, so these are the pathogenic cells that had been killed by heat. Okay, and these cells that had been killed by heat, they did not do anything to the mice. The mice did not get sick. And his next step then was to take some of those heat-killed cells and mix them with the living R cells. Remember, the R cells were the ones that were non-pathogenic. So he took some of these dead, potentially pathogenic bacteria and mixed them with the living non-pathogenic bacteria. And the result here was that the mice would die. They would get pneumonia and they would die. And so when they took a blood sample from the mice, they were found to have living S cells in them when they had been injected with heat-killed S cells but living R cells. And so Griffith was then able to conclude that bacteria had incorporated some kind of hereditary factor. Okay, so transformation was occurring, where these bacteria were picking up hereditary factors from an outside source. So in this instance here, these R cells were picking up DNA from these heat-killed S cells. He did not yet know, though, that it was DNA. So our next experiment here was done by Avery, and he was working with MacLeod and McCarty. And so this particular experiment took Griffith's experiments a step further. So these were a continuation of an extension of Griffith's work with the S cells and the R cells, and they were able to prove that it was actually DNA that contained the heritable information. So what they did is they isolated all these different parts of the bacterial cell. So they isolated all those different parts of the bacterial cell of the pathogenic S strain cells and introduced the different parts to the R strain, and they found that the only ones that converted that were able to uptake this hereditary factor were the ones that had been exposed to the DNA. So they were then able to conclude and take Griffith's experiment that extra step and conclude that it was actually DNA that carried the heritable information. So our next experiment here is Hershey-Chase. And I think on your handout there's a typo. I think it says Hershey. Okay, but Hershey-Chase. And they worked with viruses, and they were able to demonstrate again that the genetic material is DNA. So they basically did two trials. Okay, so they had their first trial, or their first batch here, where they had mixed radioactive proteins into the viruses. And so they're colored pink here with these radioactive proteins. And so they incorporated radioactive markers into the proteins of the virus, allowed the virus to then infect the bacterial cells, and then they would centrifuge or separate them out. And when they separated out the now infected bacteria in the virus portions, they were able to see that those radioactive protein markers were not in the bacteria. They were still in the virus. So it helped to eliminate proteins as the heritable factor. Okay. Their other trial, so their second trial, they inserted radioactive phosphorus. Remember, DNA contains a sugar phosphate and a nitrogen base. And so this radioactive phosphorus was able to mark the DNA. So in this instance now it's blue. Okay, and so again, they allowed the viruses to infect the bacteria, centrifuge, separate them out, and they see that the bacteria now has radioactive markers on it, not the viruses. So they were, again, able to prove and determine that DNA is the heritable factor here. Okay, so our last experiment that we're going to review today is Chargaff's. Okay, this is the base pairing rules. Okay, so Chargaff used what's called paper chromatography. Basically separating out pigments. Okay, so he used paper chromatography and an ultraviolet spectrum to be able, spectrophotometer, to be able to determine these base pairing rules. And so what he was able to determine, what he was able to demonstrate is that organisms have approximately the same number of guanine and cytosine, and then the same number of adenine and thymine. And so he was also able to determine that these percentages were going to be unique to various species. Okay? That as you can see here, all these different species have different amounts of adenine, thymine, as well as different amounts of cytosine and guanine. Okay, so let's do a little DNA structure review then. So if y'all remember, your monomer of DNA is going to be your nucleotide. Remember, your monomer is your building blocks. So DNA is made up of long chains of all these nucleotides strung together. So your nucleotide is going to consist of a phosphate group. Okay, so every nucleotide will have this phosphate group. It will have the sugar, and in the case of DNA, it's going to be deoxyribose sugar. As you can see here, compared to ribose sugar, deoxyribose sugar has one less OH group on it than the ribose sugar does. Okay, so I've got my phosphate here, I've got my deoxyribose sugar, and then I've got my nitrogen base here. So let's take a look first at the sugar. Okay, so the carbons in the sugar, remember sugars are usually found in a ring shape, as you can see here, and the carbons in these sugars are going to be numbered. Okay, and they're numbered, you can see the little numbers here. Okay, let's get rid of that. Okay. So you can see I've started here with carbon number 1, and it will go around in a circle like this. Okay, and so it's a 5-carbon sugar, so I've got 1, 2, 3, 4, and 5. And some of these carbons are significant. Carbon number 1, okay, right here, carbon number 1 is where the nitrogen base is attached. Okay, carbon number 2 is where I get my difference between the two sugars, between deoxyribose and ribose sugar. Carbon number 3 is where the next nucleotide will attach. And that becomes extremely important in a little bit when we're talking about replication. So this carbon down here in the corner is where the next nucleotide will attach. And then carbon number 5 out here, that's where the phosphate group will be able to attach to it. And this gives, I don't know if you guys remember this from your freshman year, this gives the strand of DNA what's called a 3' end and a 5' end. And so when we're talking about 3' or 5', we're talking about basically which carbon is available, which carbon is on the end. Is it the 3' carbon or is it the number 5 carbon? Okay, and we'll look at that in just again some more in just a minute where we look at full strands here. Okay, so let's review a little bit then about our nitrogen bases. So remember there are 4 nitrogen bases, right? We've got adenine, guanine, cytosine, and thymine. We are not focused on uracil at all right now, okay, because we're not talking about RNA right now at all. We're going to talk about DNA and DNA replication, and we'll get to protein synthesis in 2 weeks or so. Okay, so there are basically two kinds of nitrogen bases. You can have either a purine or a pyrimidine. And if you look at them structurally, the purines have 2 rings, where the pyrimidines only have 1. Okay, if you guys remember this phrase from your freshman year, to Remember your pyrimidines are cut from purines. And that's not literal. It's not like I took this 2-ring purine and I cut it in half and now I have a pyrimidine. Okay, that's not what happens. But it does allow you hopefully to remember, okay, that my pyrimidines are 1-ring structure. My purines are 2-ring structures. I also think about it too that the longer the word, the smaller the structure is. Okay, with my purines, the shorter the word, the bigger the structure is. So my pyrimidines are cut from purines, and you'll notice the word cut is capitalized. Okay, so pyrimidines are cut. Pyrimidines are cytosine, uracil, thymine from purines, and so that leaves adenine and guanine to be our purines. Okay, so looking at the DNA strand itself, remember DNA is a double helix, that twisted ladder. Okay, so it's a double helix. It has the strands on it will run what's called anti-parallel. So if you look over here, you can see on this strand on the left, the 5' end is available on the top and the 3' end is available on the bottom. All right, so this one is said to be running from the 5 to 3 direction. Okay, and over here on the right side, okay, it's running 3 to 5. See on the top, the 3 is available and the 5 is available on the bottom there. So they run what's called anti-parallel, or essentially opposite to one another. Okay. The outside, remember the outside of our DNA double helix is that alternating sugar phosphate backbone. You can see it here, the alternating blue and yellow sugar phosphate backbone. You can see here as it's unwound, I've got a nucleotide, a whole bunch of nucleotides, all of these little sugar phosphate nitrogen bases held together. So there's my monomers. And then I have that alternating sugar phosphate backbone on both sides of the DNA strand. And those nitrogen bases then will run right through the middle. Okay? Remember our nitrogen base pairing rules. Okay? We've got a purine and a pyrimidine that have to go together. Okay? If you look over here on the right, you can see that there are, if you tried to put 2 purines together, they'd be too big and they wouldn't fit in the space. Okay? If you tried to put 2 pyrimidines together, they'd be too far apart. They wouldn't be able to bond. So it was left with, it had to be a purine and a pyrimidine. Okay? And so adenine, they're held together. Those nitrogen bases are held together by hydrogen bonds. If you remember from when we talked about water, hydrogen bonds were those relatively weak bonds. Okay? But remember, there are millions, billions, up to like 3 billion base pairs in each of your cells. So if you've got billions of these hydrogen bonds, then collectively they'll be pretty strong. So adenine and thymine, when they bond, they only form 2 hydrogen bonds. Cytosine and guanine will form 3. But you'll notice again you've got a purine and a pyrimidine paired together all the way through. Those are again based off of Chargaff's rules too as well, that adenine and thymine and cytosine and guanine will go together. Okay, so as a general rule, so in humans, the DNA is going to be in the chromatin form. Okay, the chromatin is that loose, not overly tightly coiled form, okay, except for when the cell goes through mitosis, and then it will condense into chromosomes. So you only see those chromosomes when the cell is preparing to go through mitosis, and basically that's extra coiling of the chromatin state. Okay, so in eukaryotes, so in cells that have organelles, so in eukaryotes, the DNA will be wrapped around proteins called histones. And that helps to condense that DNA since there's so much of it in each of the cells. So in eukaryotic cells, the linear DNA is wrapped around histone proteins, and they form what are called nucleosomes. So that nucleosome is a combination of the DNA wrapped around the histone, and those will be in eukaryotic linear DNA only. Prokaryotes, on the other hand, they don't have linear DNA. Prokaryotes have circular DNA, and so they won't wrap around histones, and it won't form the nucleosomes because, again, they have circular DNA as opposed to this very long linear DNA. So when prokaryotic DNA, for example, like this E. coli here, so when prokaryotic DNA it needs to replicate, the chromosomes will attach to the plasma membrane at what's called the origin of replication. Okay, so the DNA attaches to the plasma membrane. This becomes the origin of replication, and then it will start. You can see where it's being pulled apart here. Okay, it's being separated out here, and it will replicate in both directions. It will replicate this way around the circle as well as this way around the circle to help speed the process up. Prokaryotic DNA replication is significantly faster than eukaryotic because it's a lot smaller. They only have about 4.6 million base pairs, whereas eukaryotic cells, like I said, will have about 3 billion base pairs. Okay, so prokaryotic DNA replication, like I said, we're not going to focus on it really. I just need you to know that it attaches to the plasma membrane, forms that origin of replication, and it's replicated in both directions. So eukaryotic DNA is going to be a little bit different because eukaryotic DNA is so much bigger than prokaryotic DNA. So eukaryotic DNA will have multiple origins. You can see on this strand of DNA that we have here, okay, there are, just in this little section, there are 3 different origins that replication is starting from. And all of these will occur simultaneously, and it will go, again, like it did in prokaryotic DNA replication, in both directions. So eventually these two will meet up, and it will help speed the process up of copying this very long linear chain of DNA. So we've got a couple enzymes that we need to keep in mind and remember as we're going through this. So our first enzyme that we have here is helicase. Okay, helicase's job, hopefully the ACE lets you know it's an enzyme, and the helicase is going to open up the helix. The helicase enzyme is going to go through and it's going to break those hydrogen bonds. Remember, hydrogen bonds are not very strong, so they'll be relatively easy to break, and it will break those hydrogen bonds to separate the strands. And so this is where you're going to get all of these origins of replication and these replication forks. Okay, right here, where it has broken the bond there, that's going to be what's called a replication fork. And so the helicase enzyme is going to help create those by opening up the DNA. The problem you have is this DNA is wound so tightly that as the helicase comes through and is opening up the DNA, it will actually start to wind this even tighter. Okay? And so as that's winding tighter and tighter, that could possibly damage the DNA. So what we have now then is another enzyme called topoisomerase. Okay? To me this picture looks kind of blurry, so I'm going to rewrite some of these a little bigger so you can see them a little bit better. Okay, so we've got another enzyme here called topoisomerase, and that topoisomerase is going to help stabilize that DNA there. It's going to help relieve the stress from unwinding the DNA. It may snip one or both of the strands of DNA, and then it will splice them back together to help relieve some of that tension there. But it's going to help keep the DNA from being damaged due to the DNA unwinding from the helicase. So now that we've opened up this DNA strand, we need to now add some nucleotides to it. So both sides of the DNA strand, so both sides of the DNA are going to be available for copying, and nucleotides then would need to be added to both of them. So the nucleotide that is not attached yet, that is actually going to have 3 phosphate groups on it, as opposed to just one. It actually is free-floating with 3 phosphates, so it's a triphosphate, and as it joins, it will cleave off 2 of those phosphates. Those 2 phosphates are what's called a pyrophosphate, and those will be recycled. One of the key things that we need to always remember about nucleotides is they are always added in the 5 to 3 prime direction. Okay, always, always, always added in the 5 to 3 prime direction. So what that means, to simplify that even further, is they are only added on this 3 prime end. Okay, you cannot add a nucleotide onto that 5 prime end. If you remember, the 5th carbon was the one where the phosphate was attached, so that would be up here. Okay, this one cannot get a new nucleotide added to it. Okay, it has to happen down here on that 3rd carbon on that 3 prime end. So the enzyme that's going to help add these nucleotides onto the strand, onto the new strand that's forming, is what's called DNA polymerase. And there's a couple of different kinds of DNA polymerase, but you don't need to know, you don't have to be able to distinguish between the 2 kinds, or the multiple kinds. So DNA polymerase, one of the DNA polymerases will correctly pair up the bases. Okay, so you can see that happening right here. It's bringing in the correct nucleotide. If there's an adenine, it's bringing in a thymine. If there's a cytosine, it's bringing in a guanine. Okay, it also helps ensure the formation of a covalent bond to the sugar. Remember, the covalent bond is the strong bond, much stronger than a hydrogen bond, and it will help ensure that there's a covalent bond formed between these 2 nucleotides, where there's just the hydrogen bond there between the nitrogen bases. But we need the covalent bond, okay, as the chain lengthens. We also have another type of DNA polymerase that's going to remove the primer. So when this DNA starts to form, it needs to be initiated with an RNA primer. Okay, so this RNA primer is going to signify the start of the DNA replication. Well, we're not making RNA, we're making DNA. So we've got this primase enzyme, you can see there. Okay, so we've got this primase enzyme that's going to lay down this RNA primer, and that initiates the formation, the beginning of the DNA replication. It really starts the process there. Okay, but when it's done, we need to replace that RNA primer with DNA nucleotides. And so another form of polymerase will come in and replace that RNA primer with the appropriate DNA nucleotides. And so, as I mentioned before, both strands, so you can see original strand here, original strand here, both strands are going to act as a template. And if you remember back to earlier where we told you could only add onto that 3' end, that's going to be a problem because they run anti-parallel. So we need to address how that is dealt with. So the 2 strands of DNA get termed the leading and what's called the leading and the lagging strand. Okay, so you can see we've got leading and lagging strand, and it depends on what direction it's going. So in one direction, it's going to be able to be synthesized continuously. It's not a problem. Okay, here's my 3' end. So there's my 3' end, which means my other end is going to run, so this side runs 3 to 5, which means my opposing end runs anti-parallel, so it's going to run 5 to 3. So remember, I can add onto this 3' end. So this DNA polymerase can just keep adding and adding and adding and adding and adding all the way down. That's not a problem. So that'll be synthesized in one long continuous piece. The other side, though, is what's called our lagging strand. And so with the lagging strand, it has to be synthesized in pieces or fragments. So in the lagging strand, my origin of replication would have been way over here. Okay, and so this, as it heads this way, I've got a 5' to 3' end, which means my new strand, my anti-parallel strand, is going to be 3' to 5'. Well, I'm replicating. I'm laying down new nucleotides way down here. Okay, well, the problem on my lagging strand is down here is my 5' end, and I cannot add to that. So basically the DNA has to wait until it is opened far enough so that it can make a fragment. And it will make fragments and synthesize it in this direction. So it'll make that short little fragment. It'll wait for it to open up again. So it opened up far ', it made a fragment. It opened up a little further. It was able to synthesize another fragment, all going this direction, because you can only add on that 3' end. And so that's what's called the lagging strand. And these little fragments are called Okazaki fragments, named again for the person that found them. So these little fragments will eventually, they will run into each other. You know, as this fragment here, let's erase some of this mess here. So as these fragments are being synthesized, you can see how they will start to run into one another. And as they run into one another, the RNA primers will get replaced. Remember that DNA polymerase will come in and it will replace these RNA primers. And then it will essentially splice or bring together all these little small DNA strands, these Okazaki fragments. And this is done by an enzyme called ligase. So ligase is going to tie together all those little Okazaki fragments into one continuous strand, and those will be held together by covalent bonds. So this form of replication is what's called the semiconservative model. And this was talked about a little bit your freshman year. This semi-conservative replication. Remember, you conserve something, you save it. And so with the semi-conservative form of DNA replication, every time when the two DNA strands separate and they form the two new complete DNA molecules, each one consists of half of the original strand, so it's saved half, remember semi is half, so it's saved half of the original strand. And the other half of it is the new strand that was synthesized. So this is this semi-conservative model of DNA replication. Okay, so there's not a lot that you need to know here. What I really just want you to be familiar with it. So if you see some of these terms pop up or anything, you will know what they're talking about. So one of the things we need to keep in mind is I told you you've got these RNA primers, right? So there's an RNA primer at the beginning of our strand. Okay, and this strand is going to be my leading strand, right? It's being synthesized here in this 3' direction, and I can easily add onto the 3' end. Okay, and then on my lagging strand, I also have those RNA primers. Well, I can replace these RNA primers on the lagging strand without a problem, right? Because I'm being synthesized now in this direction, which is the 3' end, so I can just add on to that 3' end, and I can replace that RNA primer. On my leading strand, however, when that RNA primer is removed, that is the 5' end of the strand, and you cannot add any nucleotides to that 5' end of the strand. And so over time, that would end up shortening the DNA, because you've got this gap that needs to be filled, and so basically it just gets cut off. And so it ends up shortening the DNA. And so to prevent this, our DNA has on it what are called telomeres. And so a telomere is just basically a repetitive DNA sequence. It's essentially useless. So it's this repetitive DNA sequence that takes into account that it gets shorter every single time the DNA is replicated. Okay, so every single time that DNA is replicated or copied, it gets shorter because you can't replace that primer on the leading strand, on the 5' end. And so our DNA has these built-in useless repetitive sequences of DNA that if they get cut off, it doesn't matter. Okay? This shortening of the DNA is one of the things that's associated with the aging process. In certain cells, we have an enzyme called telomerase, and that telomerase enzyme is going to actually add on to the telomere length. This would happen in gametes because you can't afford to have the DNA be shortened in growing and developing gametes as well as children. You'll find this in blood cells, epithelial cells, Cancer cells, they'll have a high presence of this telomerase enzyme. Cells that seem to live forever and not die, they potentially have this telomerase enzyme in them, so you don't see the effects of that DNA shortening. The last thing I want to touch on is if the DNA essentially gets a little bit messed up when it is going through this replication process. And I don't necessarily mean, we're not talking about mutations here. We're talking about little small fixes that the cell itself may be able to fix and repair. Okay, so you may have the possibility of a distortion. This one here is sorry, I forget sometimes. This one here is what's called a thymine dimer. Okay, that's usually caused by UV exposure. So that is something that can cause this DNA to be damaged. Chemicals, can't spell. Chemicals, UV light, X-rays, things like cigarette smoke, all of these things can damage the DNA. And so your body the cells do have a limited amount of repair that they can do to the DNA. So again, we've got these DNA polymerases that will come into play. You've got DNA polymerases that can proofread and try to replace incorrectly paired nucleotides before anything ever even gets essentially finalized. You have another enzyme, okay, that's called nuclease. Okay, nuclease is a repair enzyme that will come in and essentially cut out the broken part. You can see it here. Okay, so here's our damage. The nuclease comes in, it cuts out the broken portion. The polymerase can replace it with correct nucleotides there, and then the ligase will then seal it back up. Okay, just like it sealed those Okazaki fragments together. So the cells do have some limited repair abilities for their DNA. So we're going to be working with replication next class, and then we'll be moving on to why it's going to need to replicate the DNA in preparation for doing mitosis.